welcome back. This is one of the most exciting parts of the CHAOS workshop because we're going to hear really from the next generation of scientists who are each going to be presenting 12-minute talks. These were the poster award winners, and just for a little context on process, so when all of the abstracts come in, they're blinded and all author information is taken off and they're distributed among the organizers who, 2 or 3 of whom each rate each poster, again blinded, and so we're very excited to be able to offer travel prizes and the recognition, and hopefully really looking forward to everybody's presentation. So there's going to be 12 minutes for every talk. There'll be 3 minutes for questions, so keep your questions short. And we're going to try to keep this moving along, and so we're going to change speakers probably even during the question period. Okay? After the talks, we'll ask all of the speakers to stay, and we have certificates for everybody, and we'll make sure that we get a group picture of everybody up front. All right, take it away.
Does this work? Yes. All right, well, thank you very much for the opportunity to present my work. This is a project that is part of my PhD, and it's a collaboration between 𝗤𝗏𝗵𝗵𝗵 and 𝗤𝗏𝗺𝗵𝗵𝗵𝗏. So if you look at this very, sorry? This is the slide. But thank you. So if you look at this somewhat Microsoft background-looking image, the information that actually gets to your brain looks a little something like this. So we have this very high acuity fovea in the center of the visual field, and then the degraded periphery outside. Of course, this is simplified, but broadly, this is what it looks like. And because of that, as soon as you look at this image, you will start moving your eyes around to different objects on the screen that are of interest to you to really absorb as much information as you can. But because you don't just move your fovea, you move the whole eye, the information that actually is on your retina looks more like this. So every time you move your eye, the visual field completely shifts and you get different information. But even though that's the case, we don't get whiplash every time we move our eyes. We have a very continuous perception of the visual environment. And one hypothesis for why that might be, that was put forward by Martin Rolfs and his lab, is that your fovea actually predicts incoming information. So that if you plan a saccade to an object, so to this deer, for example, the information is actually fed back from higher areas to the foveal retinotopic cortex, so that when you execute the saccade, these representations can align, and it's a more smooth representation. And we wanted to test this hypothesis in fMRI. So our research questions were, can we find this foveal feedback in the early visual cortex, or in the foveal parts of the early visual cortex? What information specifically is fed back if we find this activation? And then lastly, which brain regions might be involved in driving this? So potentially, where does this information come from in the higher in the higher cortices? And to assess these hypotheses, we designed a combined eye tracking and fMRI experiment where we used a gaze contingent design to get at this question. And I'm going to take you through the design real quick. So this is the experimental condition. The participants have this fixation point on the screen, and then after a few seconds, an object appears. The participants are supposed to perform a saccade towards this object, and as they do this, the object disappears. And then it reappears again, saccade, and it disappears over and over in an experimental block. And the reason why we do this is we want to capture sort of the saccade preparation towards this object, but we never want the object to appear in the fovea directly. Because our question is, can we decode these saccade targets from the foveal parts of the early visual cortex, even though it never appears in the fovea directly? We also had a control condition where we did the same thing, but everything happened in the fovea directly. So that was the presentation of the stimulus over and over with the same frequency or the same timing as in the experimental condition. And here we really wanted to test, can we also cross the code? So can we cross the code from experimental to control trials if we find this feedback to see, is the representation we find in the experimental condition similar to the one we find in this control condition? The stimuli that we used were these 4 natural images, and we matched them sort of similar to what Stefania showed. That was the inspiration for this in terms of categories. So we had 2 different categories. We had the animals on top and then the instruments on the bottom. And we had different shapes. We had a more horizontal and a more vertical shape to sort of assess, can we find category or shape-specific information in the foveal area? So is it more higher level or just lower level information that we find? Now let's go to the results. We used the support vector machine decoding, and we used five-fold cross-validation across runs, and we always did pairwise decoding between these stimuli that we used. And first of all, let's start with the decoding from foveal V1. As you might expect, in the control condition, the decoding was fairly good because the object was presented directly in the fovea. But interestingly, also in the experimental condition, we could decode these stimuli, which is really cool because that means that somehow, even though the object is never presented in the fovea, the information gets fed back to these foveal areas. And because we could decode it also means that the activation we find is somewhat stimulus content specific. Next up, as I said, we also wanted to cross-decode from experimental to control trials, and we see that also this works. So it seems like the activation that we find here, or the representation, is kind of similar across direct representation and this feedback that we find. Next up, we wanted to look a bit more at the content, so the information that we find in this decoding. And for that, as I said, we have these 4 images. For this room, probably 4 images is not a lot, so I don't want to overstate what we found. But we use these images to sort of compare what is what is the shape content versus the categorical content of this coding that we find. So we first did this sort of baseline decoding where we always compared the 2 stimuli that are different in terms of semantic category and in terms of shape to see how good is the decoder if we give it really all the information that we have here, the categorical and the shape information. Then we systematically took away information. So we first compared within shape. So here we drastically reduced the shape information that we give the decoder. And we see that in the control condition as well as in the experimental condition, the decoding accuracy drops. So it seems like shape is somewhat represented in the cortical or in the neural code that we were capturing here. We then did the same thing within categories. So here we took away the categorical information to see, does that maybe also degrade the decoding somewhat? And we don't find that in the control condition and also not in the experimental condition. So again, these are only 4 stimuli, but it seems like the foveal feedback activation that we find really seems to code for visual shape information, and there doesn't really seem to be higher order categorical information that is fed back to the fovea. Just as a control, we did the same thing also in the lateral occipital area because we know there's categorical information encoded here. And here we find that this pattern is reversed. So we see that there is these objects that we chose do capture the categorical information, it's just not visible in foveal V1. Next up, we wanted to see where in the visual field can we find this information. So we decoded not just from the fovea, but we decoded from the very central part of the fovea all the way to the outer parts of the periphery we could capture. And in the control condition, as you might expect, the decoding is really good in the fovea, and then it kind of goes down towards the periphery because the stimulus wasn't presented there. But in the experimental condition, we find this nice U shape, which also makes a lot of sense because the periphery is where we present the object, so this is where we can decode. Then it gets sort of worse as we move towards the fovea where the stimulus disappears, and then we see the specific feedback activation in the fovea directly. And that was really nice because it also showed us that it's not some sort of overlap from the periphery that we can decode from the fovea, because then you would sort of expect more of a straight line from periphery to fovea, but it's more of a specific feedback activation that seems to be distinct from the bottom-up peripheral decoding that we find. Now lastly, we also wanted to see which cortical area might be related to this, which might drive this or initiate this in some way. So we looked at the decision scores of the foveal decoding compared to the peripheral decoding. And we did a parametric modulation analysis to see, are there areas in the brain that correlate with either good foveal decoding or with good peripheral decoding, sort of as a proxy of this bottom-up versus top-down processing that we find. And for that, we used Neurosynth to do an RI analysis, because our hypothesis was it should be some eye tracking or eye movement related area that is involved here. So we used Neurosynth to kind of isolate the most common eye movement related areas. And that gave us the frontal eye fields, the interparietal sulcus, and the lateral occipital areas that have in some way been associated with eye movements. And here we found that they're all associated with foveal as well as peripheral decoding. But especially the IPS seems to be specifically more activated for foveal compared to peripheral decoding. So we thought that this could be at least a plausible region as a booster or a driver of this foveal feedback. Even though, of course, this is correlational, so we don't know does it actually originate there, but we know it's probably involved in some way. So just to summarize, we found that we can decode peripheral Saccade targets from the foveal parts of V1 and also V2 and V3 actually. We found that this foveal feedback activation is stimulus content specific. We also found that it seems to resemble the activation elicited by direct presentation. And we find that it seems to code mostly for low to mid-level visual information, so shape information in this case, and we didn't find any categorical information that may be fed back. And lastly, we find that the interparietal sulcus may act as some sort of booster for foveal feedback. We also have a preprint out that is now under review at eLife, if you have any more, if you want to read into it a bit more. And with that, I want to thank my collaborators and, of course, my group in Gießen and in Leipzig and the funders of this research. And thanks to you all for listening.
Questions? Back there, and then Angelica.
Hi, thank you very much. Sorry. I was curious how you managed to time lock when the stimulus is represented in the periphery of the field with respect to, so it should happen earlier with respect to when it's encoded in the fovea. So I was curious because a Saccade is very quick and the bold is very slow instead.
Yeah, so we used a block design. So our idea was that we capture this process sort of over and over again, so that over time we can, this really drives the bold signal sort of over time. And that's also why we made sure that the object never appears in the fovea directly, because that would have sort of contaminated the block, I guess. So this is exactly why, because we don't have this temporal information, that we really wanted to make sure that the object never appears in the fovea. And we also did some processing afterwards to make sure, and we excluded all the blocks where at least there was a doubt that maybe the object could have been in the fovea for even a millisecond. We excluded all those blocks and only used those where we were sure the object was never in the fovea directly.
I'm curious if you've tested any sort of control condition where people do not make a saccade, but that same visual information is presented in the periphery.
Yeah, we did not make that control condition, but other people have done that. So there was this Williams study in 2008 that did the same thing, and they also had a control condition where there was no task and no saccade and nothing, and they couldn't decode it. And also 𝗏𝗏𝗏 et al. Did a study from 2016 where they also had this control condition where they found nothing, so that's why we then decided to have more experimental trials and not include this control condition.
Thanks.
Thank you. I have a question regarding the last analysis you presented within the regions you identified using Neurosynth. That's nice, it fits with the rest of your results, but wouldn't you, in order to break this prediction, also have to show some areas that are not related to eye movements and show that this no longer holds?
What do you mean that breaks it?
Well, you're only showing areas that are compatible with your hypothesis, but you would also have to test areas where you would predict that this should not be there and demonstrate it.
You mean as a control where we say, okay, this is where there should not be any activation to make sure? Yeah, that's a good point. Mm-hmm. Yeah, thank you.
Any other questions? We have time for one more, if there's another one. No? All right, well, thank you very much.
Is it you now?
So, hello everybody. First of all, I would like to thank the organizers for this good conference and for this great opportunity. So I'm Sarah, and when I was a child, a very surprising phenomenon was discovered. We tend to imitate in our brain the body movements that we see, and this has been referred to as motor simulation. Soon after this discovery, motor simulation has been suggested to contribute to our ability to perceive, interpret others people's actions. For instance, the body movements or facial expressions. Now, I'm 28 years old, and it still remains unclear whether, and if so, motor simulation does contribute to these abilities. So this is what the studies I'm going to present are about. So in particular today, I will focus on the question whether motor simulation contributes to our ability to recognize facial expressions like these ones. But of course, before me, others group of researchers have already tried to contribute to this question by testing the ability to recognize facial expressions by people born with a congenital facial paralysis. So due to an absence of the facial nerve, these people cannot contract their facial muscles. And here, for instance, 𝗤𝗧 is giving you more information about this syndrome that is called the Moebius syndrome.
I was born with a really rare neurological disorder called Moebius syndrome. This syndrome is so rare that it only affects one person in every 3 to 4 million. And this syndrome affects my 6th and 7th cranial nerves.
So we know from previous studies that these individuals, like 𝗤𝗧, who cannot overtly or covertly imitate facial expression, were nevertheless able to recognize facial expressions with typical speed or accuracy. However, being efficient at something is not only being able to be fast, to be accurate, but it also requires to do it with minimal effort. However, it's still unclear if motor simulation does contribute to this ability. So, about this point, some researchers have been suggested that motor simulation does contribute to do that. For instance, Rizzolatty and Sinigaglia proposed that motor simulation is the only way to recognize actions, but it allows to do it without any effort. So we tested that hypothesis. So to do so, we recruited 16 individuals with the Moebius syndrome, and to evaluate their ability to imitate facial expressions, we assessed their ability to execute facial movements involved in the production of the 6 primary facial expressions. So joy, sadness, anger, disgust, surprise, and fear. And here in the y-axis, you can see the different facial movements that we asked them to try to perform. And the x-axis corresponds to the 16 individuals with Moebius syndrome. The ability to execute each facial movement was then categorized into one of the 4 categories. So absent when no movement was detected, very limited when only minimal movement was observed, like a few millimeters, limited when the movement was not complete, but more marked than the very limited ones, and complete when the facial movement was really marked bilaterally. And as you can see in the matrix, most of these people could barely execute any of these facial movements. Then we asked them to perform a dual task, and here is how it looks. First, they saw a fixation cross, and then they heard a sound, an emotional sound, and at the same time they saw a face. And we asked them to do 2 things. First, to categorize the emotional sounds as fast and as accurately as possible. And second, to look to the screen to be able to remember the identity of the actor. So this experiment included 2 blocks. For the first one, contrasted fear versus anger. The second one, sadness versus disgust. And all the sounds were presented twice, once in the congruent condition and once in the incongruent condition. So incongruent means that the facial expression didn't match the emotion conveyed in the audio stimuli. So here, with this task, we never asked them to process the facial expression presented on the screen. This information was irrelevant, and we asked them to do 2 demanding tasks. So we assume that if motor simulation contributes to, so therefore we assume that if we observe a congruency effect in this task, it will reflect an automatic, effortless recognition of the facial expressions. So the critical question was then, can we detect a congruency effect for the individuals with the Moebius syndrome? So, of course, at the end of the dual task, all participants completed categorized the sounds in isolation to make sure that they were able to recognize the emotional sounds without the help of the facial expressions. And this was the case, so let's move on to the result of the dual task. So, in the first graph, in the y-axis, shows the number of correct responses for the congruent in light gray and incongruent condition in dark gray for all participants. We order participants from left to right with the ones who showed a larger difference in accuracy between both conditions. And all the asterisks on top correspond to a significant congruency effect assessed with exact McNemar tests. So they answered more accurately to categorize the sounds when the facial expression was congruent. Below, participants are aligned as above, and this graph displays the mean response times for each participant at the task for the two conditions. And for the response time analysis, we conducted per t-test. But what is important here is that, as you can see in green ink in both graphs, 13 out of the 16 individuals with Moebius syndrome showed a significant congruency effect on at least one of the two variables. And when we conduct Bayesian group analysis, we find that the strength of the evidence in favor of a larger congruency effect for the control group is considerably weaker than for the alternative, that for H0, whatever the width of the prior. So in our opinion, these results show that motor simulation is not necessary to effortless automatic facial expression recognition. However, of course, we need to keep in mind that the stimuli that we used in this study were very prototypical, so very easy to recognize. Maybe these results may not generalize to other kinds of stimuli, for instance, when the facial expressions are very ambiguous or less familiar. And this is in line with the hypothesis that there is two main pathways to recognize actions. So when the observed body movements are very familiar, presented in optimal viewing condition, like in the experiment that I just showed you, the results of the visual perceptual analysis of the actor's body postures and movements may be matched easily and rapidly onto a corresponding representation in visual memory, allowing for fast, easy, almost immediate recognition of the action. However, if the observed body movements are unfamiliar or familiar but presented in a very unfamiliar format, such as point lights, the results of the visual perceptual analysis may be too different from the visual representation stored in memory. So in this case, this model assumes that if the first route fails, then motor simulation could contribute to action recognition. So here for this talk, I don't want to enter into too many details here, but the idea is that motor simulation could contribute because it allows observers to retrieve information about what action this movement allowed them to produce when they carry them out, or because they allow them to maintain the perceived body postures and movements in memory. So we tested that hypothesis, and just to tease you for the last minute of my presentation, here's what we did. So we asked individuals with Moebius syndrome who cannot covertly imitate facial movements to recognize facial expression depicted into point light display, as well as video of action that served as control condition. And the result in this task showed a significant interaction effect. So the individuals with Moebius syndrome were significantly less good, less accurate than controls to recognize facial expressions than actions. So here, the take-home message is this. First, motor simulation seems to not be necessary to efficient recognition of the facial expressions, at least when they are very easy to recognize. And second, motor simulation might still contribute for you and for me to help us to recognize facial expressions, but precisely how and when remains to be clarified. So let's just hope we get the answers before I turn 30. So here I would like my supervisor, my colleagues, all the participants, and a special thank for the individuals with the Moebius syndrome for their trust, their time, and availability. Thank you very much.
Laurel has the first question.
Great talk, really interesting study, thank you. I have a question, though, about your use of the term motor simulation, since as far as I heard, you didn't really measure simulation capacity. Instead, you just, I think, assumed that people with facial paralysis cannot simulate internally facial expressions. And I wondered if there would be a way to more directly test your hypothesis by including some kind of task with which you could more directly measure motor simulation. But simulation doesn't necessitate the enactment of an actual motor activity, right? There's something prior to that stage of processing, right, where you have some kind of more abstract representation of what the movement could look and feel like, which is not a motor program per se. So, and you know, for example, hand rotation tasks are used where the degree of deviation between what you perceive, what you see, and where your own body is actually creates an RT effect. The further your own hand is from you know, you're trying to do left or right discrimination. I don't know if there's something like that for facial simulation, but something like that would be more convincing to me. So what are your thoughts about that?
So you're asking if motor simulation does exist for these individuals then, in another way? So to answer to that question, I don't know if there is an alternative to do simulation of simulation of an observed action or facial expression. I don't know if there is another way. Here, the precise question was about if we don't have the ability to execute ourselves movements, is it impacted the way we can recognize facial expression or action, for instance?
Sure. You know, forgive me, that is a little different than the way you posed it.
Okay.
You use the word simulation.
Yeah, okay. So, yeah, oh, sorry.
That was great, thank you. On the very last slide you showed with the point light walkers and point light expressions, I think I missed your interpretation of why the IMS group is also impaired at the point light actions.
Yeah, so I didn't explain that, but with these individuals, they also shown visual deficits. So it might explain why, even in the control condition, they are less good than controls. But the information, what is more important here, is that we observe an interaction effect. So even if they have problem to perceive action in general, let's say, the deficit is more marked when they have to recognize facial expression. So this was the idea with this experiment. Thank you.
All right. Thank you so much.
All right. Thank you. Well, first of all, I would like to thank you everyone to be here and the scientific committee for their consideration. Today, I'll talk to you about my current research project, including 2 fMRI multibox pattern analysis studies. This research project contextualizes in a broad research field using neuroimaging techniques, and in particular fMRI, in investigating movement planning, namely the contribution of several sub-processes such as action selection, movement, and movement preparation prior to movement execution. By employing fMRI, it has been found constantly the contribution of posterior parietal cortices, secondary and primary motor regions prior to movement execution. In particular, in a recent study by using univariate analysis, it has been shown that even for specific, very detailed movement parameters such as the grip force, it can be found both signals covariation with the intended grip force intensity prior to the application of the grip force. Usually, this kind of effects are found in contralateral brain regions, which, when localized to be in area reflecting the to-be-used body part, are considered to be effector-specific brain regions. Notably, the effect of the process of movement planning is a complex process that, as said, imply several subcomponents. What remains to debate today is how these different processes integrate together and how it is possible to first represent more abstract motor goals, encode them in effector-specific brain regions, and thereafter transform them in more specific movement plan prior to movement execution. For that reason, in the first fMRI multivoxel pattern analysis studies, we used a delayed grip force task, which allowed us not only to dissociate the movement phase from the planning phase, but also to investigate sub-processes involved during the delay. We used a parametric design in order to investigate how graded grip force intensities were stored in parametric codes prior to movement execution. More specifically, we used time-resolved decoding analysis, more specifically support vector regressor model, in order to decode for increasing grip force intensity. In the task, we used a combination of a grip force queue showing 2 of 4 possible grip force intensity and a retro queue indicating which of the 2 should be prepared in order to render orthogonal the sensory information offered to participants from the motor information they should have maintained and thereafter use for motor preparation. Thereafter, participants wait before executing the action for 9 seconds, and they executed that upon the presentation of a go cue. Thereafter, they received a feedback indicating whether they were accurate or not accurate. During the full duration of the experiment, we measured the applied force from the participants by means of an fMRI compatible grip force transducer that showed that they didn't apply force at all before motor execution, and they were pretty accurate with an overall percentage of accuracy of 67% in performing this difficult task. As said, we used a time-resolved decoding analysis, meaning that based on a finite impulse response model, as first GLM, we modeled for each time being of 1.5 seconds a single beta estimate, and one for each grip force intensity that we used to run the decoding analysis. For each time being, we ran an independent search-like decoding analysis for the full duration of the experiment. And notably, we used a support vector regression model that not only allow you to classify content-specific representation, but also to decode continuous quantities, like the grip force intensity. Regarding the results, by running t-test, we found effect in each time period we examined. As expected, during the Q period, no above-chance decoding was found due to the temporal dynamics of the both signals, while during the first delay period, we found the ventromedial prefrontal cortex. In contrast, during the second delay period, we found the contralateral intraparietal sulcus and the dorsal premotor cortex. Interestingly, when we examined the time course reflecting above-chance decoding, we found the intraparietal sulcus ramping, the prediction accuracy ramping before the dorsal premotor cortex, which in combination of an additional cross-regression decoding analysis used as generalization test suggested the contribution of IPS prior to PMD. Interestingly, we found pretty strong lateralization in the effect, but since in the task was involved just one effector, namely the right hand, we were not able to fully control for the effector-specific coding of the parametric grip force. Thereby, in a second study, we asked participants to prepare with 2 ends that will be applied force intensity. We use basically the same delay grip force task in which we just split the delay periods in 2 6-second delay periods preceded by a 0.5 effector Q presentation, asking them to prepare either with the left hand, presented in a green background, or with the right hand. In 50% of the trials, participants had to switch preparation from one end to the other end. For example, from the left end to the right end, as shown by the diagonal arrow, or vice versa. As results, basically, we use the same decoding pipelines, but in that case, we split that in two based on the hand with which participants prepared the action, and results show, consistently with the previous study, no effect during the Q period. In contrast, during the first delay period, we found systematic lateralization in contralateral EBAs and IPS. For the encoding of grief force intensities. We found that effect in the right hemisphere during preparation with the left hand, again shown on a green background, and in the left hemisphere for preparation with the right hand. In the second delay period, in contrast, we found contralateral M1s, again systematically coding the to be applied force intensities. In contrast, during the execution phase, we found a broad network spanning from the contralateral to the ipsilateral M1, showing unexpected strong effect during motor execution. Additionally, we ran two additional analyses, namely two cross-decoding analyses. The first one can be labeled a cross-and cross-decoding, in which we trained our support vector regressor on the 𝗵's reflecting preparation with the right hand, and tested that in 𝗵's reflecting preparation with the left hand, and vice versa, in order to test within each delay period whether the preparation with the two ends showed as brain correlation similar brain activation patterns. In other terms, if the grief force intensity preparation was prepared with the two end in an effector-independent code. We did the same analysis in a searchlight, but for simplicity, here you can see prediction accuracy values extracted from the peak voxel taken from the main decoding analysis, which were tested with repeated measurement t-test, Bonferroni corrected. And you can see that in no peak voxel from the main decoding analysis is shown above chance decoding. The second cross-decoding analysis can be said or called switch-no-switch decoding analysis, because in that case, we wanted to test during the second delay period whether when participants previously prepared with the same end or with the other end, for example, moving from preparation with the right hand to preparation with the left hand, we had similar brain activation codes representing the force intensities. We wanted also to see if that kind of cross-decoding were reflecting and corroborating the results of the main decoding analysis. Therefore, as before, prediction accuracy were extracted from the peak voxel identified in the main decoding analysis and tested as in the first cross-decoding. And in that case, results show a nice lateralization in the contralateral brain regions for each cued end. In the right M1, we have above chance decoding for preparation with the left hand, vice versa in the left M1 for preparation with the right hand, which confirms results from the main decoding analysis. To sum up, the first decoding analysis, the first fMRI multivoxel pattern analysis show a likely transformation process for more abstract goals or action selection in the intraparietal sulcus, and in contrast, in the second delay period, we have a more specific and detailed movement preparation in the dorsal premotor cortex. In contrast, in the second study, we explicitly split the delay period in two parts, and by explicitly asking participants to prepare one end or the other end for preparing the grief force intensity, we found systematic lateralization, which differs in terms of brain regions between the two delay periods. In contralateral EBAs and IPS during the first delay period, and in contralateral M1s during the second delay period. Again, suggesting some kind of transformation from more abstract motor information to more specific movement plans, and in that case, effector-specific coding of information, as even corroborated by cross-decoding analysis. These results are overall consistent with a two-stage action planning model, suggesting that first action is selected and thereafter prepared in motor codes. and sensor and ideomotor theories, indicating that secondary sensory regions such as EBAs and IPS could reflect the anticipation of expected sensory outcomes, which could help in a second stage in selecting and preparing actual motor plans in the primary motor cortex. Said that, I would like to thank you, the members of the Neurocomputation Neuroimaging Unit, of which I am a part, my supervisor Felix Blankenburg, my collaborator, and mainly you for your attention.
Questions? I'll get us going here while somebody's thinking of another one. I was curious, so the task obviously imposes a delay in order to be able to investigate using fMRI, which is relatively slow, this intermediate process. Do you think that intermediate process is also part of normal action when you don't have a delay imposed, or could there be a kind of motor working memory that the task imposes on the system?
I would say, on the one hand, when we want to operationalize movement planning and try to investigate the different sub-processes involved, it's necessary to separate and render the orthogonal strongly temporally correlated process like execution and preparation. On the other hand, I suspect that working memory could kick in that kind of experiment. But on the other hand, given the strong task specificity of working memory, it could even be that there is now no clear distinction between movement planning and working memory. Thereby, I would say, yes, for sure, working memory could strongly contribute in that, and with similar paradigm, indeed, it has been investigated what is called prospective motor planning. Van Ede and colleagues, Nobre, have extensively investigated that kind of process with EEG mainly. That is surprising, even because with longer delay period, you would have very nice design to investigate that kind of process. But yeah, it is even very difficult to render the orthogonal working memory process that are motor and movement planning processes.
Any other questions? All right, well, thank you so much.
Okay, so hi everyone, I'm Samantha, and the study I'm going to present today investigates how the brain processes information when we explore objects through active touch, that is, haptics. While vision is our dominant sense, especially for object recognition, touch has received less attention compared to vision in the neuroscientific research. Yet, we can often recognize objects by touch as accurately as we do by sight. In addition, there are many situations in which both systems work together to support object recognition, as illustrated, for example, in this nice painting where both seeing and touching are involved at the same time. So these observations highlight the close interplay between vision and touch for object recognition, which opens up the possibility of multimodal processes. In this regard, there are interesting neuroimaging findings showing that the early visual cortex, including primary visual cortex, V1, is involved in tactile tasks, for example, in bright reading in blind individuals, but also when sighted people haptically explore objects in the absence of vision. While these findings suggest that visual areas are involved in haptic exploration of objects, they also raise important questions. For example, what is the role of visual areas during haptic processing of objects in sighted people? Are they processing stimulus-related features, or are they supporting visual imagery processes? And that's what our study aims to explore. Specifically, we targeted the size of stimuli to address two main research questions. The first one is about whether early visual areas have a representation of the size of haptically explored stimuli, and if these could be due to visual imagery. And the second one is about whether we can also find abstract representations of size that are independent of task modality. So to address these two questions, we recruited 25 right-handed sighted people to take part in our slow event-related fMRI experiment, during which participants had to either haptically explore with the thumb and index finger of the right hand, or visually imagine one of 3 differently sized rings or circles. So we had 2 tasks and 3 sizes: small, medium, and large. Importantly, participants were blindfolded for the whole duration of the experiment, so they could never see the stimulus. The stimulus consisted of a 3D plastic tablet with 3 concentric circles engraved on its surface, and the tablet was placed above participants' abdomen while they were lying on the magnetic resonance bed. Here you can see the structure of our trials, which started with an auditory instruction indicating participants which task to perform, and also the target ring. As you can see, in order to prevent auditory instructions from conveying information about the spatial magnitude of stimuli, we didn't use words like small, medium, and large. Instead, we used letters, J, P, and Q, to be randomly associated with the 3 stimulus sizes. So we trained participants to learn the letter size associations before the onset of the experiment. After 6 seconds, they heard a stop signal cueing them to either stop haptic exploration and return to a resting position at the center of the tablet, or to stop visually imagining the circle. The next trial started after 12 seconds. So once we acquired our fMRI data, we conducted multivoxel pattern analysis in order to investigate whether we could use brain activity patterns to decode the size of haptically explored and visually imagined stimuli. So to do that, we trained an LDA classifier to discriminate between size pairs, and then tested it on a new set of data. And we did that separately for the haptic trials and the visual imagery trials. To also test for abstract representations of size, we used cross-decoding. So we trained the classifier to discriminate between size pairs in one task modality, and then tested it on the other task modality, and also vice versa. We specifically applied 𝗵𝗵𝗶 to a set of regions of interest that you can see displayed here. They are overlaid on the univariate conjunction map between haptic exploration and visual imagery. As you can see, we did not include only early visual areas like occipital pole and primary visual cortex, V1, that were the main focus of our research, but we also included regions along the ventral visual stream known to be involved in higher order visual processing of object information, including inferior temporal sulcus and lateral occipital tactile visual area, which is specifically known for its visual tactile properties. Then we also selected regions in frontal and parietal cortices known to be involved to different extents in degeneration and planning of movements, but also in imagery processes. And these include, for example, ventral and dorsal premotor areas, supplementary and presupplementary motor areas, but also primary somatomotor area and anterior and posterior intraparietal sulci. So here we can see the ROI-based results, starting with early visual areas, the focus of the research. As you can see, we found significant decoding of size during haptic exploration in all of them except in right V1. but no significant decoding of size was found during the visual imagery trials, which suggests that we can decode haptic size in early visual areas, and this doesn't seem to be due to visual imagery. This is also further supported by the significant difference in the decoding accuracies for haptic and imaging size. No significant cross-decoding was found in any of these regions. A similar pattern of results can also be found in the lateral occipital and inferior temporal areas, with the only exception that left LOTV, as you can see, also showed significant decoding of size during visual mental imagery. We can now look at the decoding results in anterior and posterior IPS, where we interestingly find not only significant decoding of size during haptic exploration, but also during visual mental imagery, and also across the two tasks, suggesting that these areas are probably storing an abstract representation of size that can be accessed from both modalities. The same pattern of results was found in bilateral dorsal premotor areas, while in the remaining frontal and frontoparietal regions, we only found successful decoding of size during haptic exploration, with the exception of left SMA, which, like left LOTV, also showed significant decoding of size during visual imagery trials, in line with the literature showing that it's involved in visual imagery tasks. Again, no significant cross-decoding was found in any of these regions. So now to sum up, we can try to answer the initial research questions. For the first question, do early visual areas have a representation of the size of haptically explored stimuli? And is this due to visual imagery? We can say now that we found haptic size decoding in early visual areas, and that this does not appear to be just due to visual imagery processes. In addition, we also found that frontal and parietal and also the ventral visual stream area show both imaging and haptic size decoding. As for the second question, we found an abstract representation of size that is independent of the task modality in frontal and parietal areas. So now let me give you the final take-home messages. So first, the visual cortex is representing the size of haptically explored stimuli despite the lack of visual information. And we think that a possible explanation for this relates to the fact that vision is our dominant sense. And we often use it to confirm what we explore through active touch, which, according to our view, makes the visual cortex a candidate region for the representation of stimulus features, with the possibility of other sensory modalities relying on this representation. As for, we also show that the ventral visual stream area, LOTV, and also SMA, show overlapping but distinct neural mechanisms for the representation of haptically explored and visual imaging sizes. And finally, we found that parietal and frontal regions, which are known to be involved in the generation of movements, encode spatial properties like size in a cross-modal format, which we think might be integrated in motor plans. In future analysis, we would also like to investigate the task-related functional connections between our regions of interest. And we also think that it would be interesting to explore whether also other stimulus-related features might be represented in early visual areas, especially during haptic exploration. And that was the end of my presentation. Thank you, everyone.
Thank you, that was lovely. Because of the nature of your task, I guess I have two questions. One is to what extent you want to call this haptic exploration. I think of exploration as maybe trying to discover something, and here there's just three, as I understand the task, 3 grip sizes that they're making.
Yeah.
And to me, that doesn't quite seem like exploration. And then related to that, then I'm starting to wonder about, do I want to think of this as visual imagery or more of maybe a motor simulation, which might then speak to the areas you found it? I could imagine if the subject was engaged in more of a haptic exploration, maybe then you would see something more parallel to visual imagery happening. So I'm just wondering why to think of this as a visual task as opposed to a motor grip task.
Mm-hmm. Well, I think that well, we did not use like a size discrimination task, which is it has already been used in a similar study where grip apertures are used to discriminate the size, but that's like a decision-making task. While here we specifically ask participants like starting from a central point, they have to move and sort of explore the target ring size by making circular movements. Okay. So in that way, I viewed our task like as an exploratory task, but because they still need to make a sort of exploration. It's not like complete exploration as you were suggesting, but I think it's still an exploration because they have to make circular movements and they have to find the right target. That's my view on that. I don't know what you think about it.
No, that helps. And then do you have any reason to think subjects are engaged in visual imagery?
Well, we assess the vividness of visual imagery using the VVIQ questionnaire, which can say something about how that correlates with neural activity. Mm-hmm. And we found that during pure visual imagery, there was a significant correlation in the left pre-SMA, which is interesting. But when we correlate the VVIQ scores during haptic exploration, which might suggest whether visual imagery is involved, we found no correlation. So I take it as a suggest. It's not, of course, evidence to exclude the role of visual imagery during haptic exploration, but I take it as a suggestion that maybe it is not involved. But that's
Thank you.
On that.
Thank you.
You're welcome.
Hi, great talk. I was wondering how you control for like whether people actually accurately pick the right circle that they're supposed to explore, and whether you can somehow make sure that they're only using like one finger on the right circle, or is there any way to control that? Yeah,
good question. We used video recording, so we could look at what participants were doing inside the scanner. And so we did not include the incorrect trials in our analysis. So yeah.
By manually excluding that, or?
Yeah, we then looked at the videos and we saw whether they were exploring the correct size. And yeah, there were trials in which they did not correctly find the target. So we had to exclude that.
Cool, thanks.
Thank you.
One more question, Wilma in the back.
Hi, great talk. Were you surprised to see a lack of ability to decode visual imagery in V1? Because there's been some prior work showing you can decode like orientations of gradings that are visually imagined from V1.
I think the literature is debating on whether visual errors are really involved in imagery processes. There's a recent review published last year which posits that probably early visual errors are not really supporting visual imagery processes, and I think, but that's my opinion, that our study is in line with that view. So yeah, I wasn't even expecting visual imagery to be involved, because that's my hypothesis, of course. So yeah, that's what I think. Yeah, I guess it's not involved, but probably it can also be further tested in future studies. That's definitely a thing.
Thanks.
Thank you.
Great, thank you so much.
Thank you.
Well, thanks a lot for for the award. And I'm Giuliano, and today I'm going to talk about this study titled Foraging in Conceptual Spaces, Neurophysiological Mechanisms of Mental Search in Semantic Memory. You already heard yesterday from Roberto about hippocampal cognitive maps and how they represent the relational structure of the environment. And here I just want to highlight the fact, this fact again, by showing that place cells and grid cells are active during navigation of both physical and more abstract conceptual spaces like social space or sound spaces. This relational structure is actually, however, just one part of the navigation behavior. Another important aspect of it is searching in both spatial and conceptual spaces. For instance, you can think of searching as animals looking for food in the environment, as an example of spatial search, but also of animals thinking and searching for ideas or concepts in their mind, as an example of conceptual search. And drawing again on this parallelism, we know from the literature that animals searching in physical space exhibit what is known as clustered exploration, meaning that they tend to switch strategies between global exploration and local exploitation of resources. So the first question we want to ask is whether we see similar strategies in conceptual spaces as it happens for spatial, in the spatial environment. And to address this question, we asked participants to perform a verbal fluency task. Verbal fluency is a very simple task in which participants are given just a category label, like animals, cities, or profession in our case. And then they have 2 minutes to think about words from that category and name them. And the idea here is that in this task, they search and use the same strategies that they use, that people tend to use during spatial exploration. And keep in mind that in this task, there is no external stimulation except for the category label. So everything is produced by the participants and at their own pace, so they can speak as soon as a new concept comes to their mind. And during the task, we recorded their voice, and we then segmented the audio recording based on word onset and transcribed the recordings as well. And as first thing, we looked at the behavioral data to characterize their behavior and see whether it matches spatial foraging strategies. And the first observation is that semantic and temporal distance, semantic distance between subsequent words and temporal distance between subsequent words are correlated. So this means that participants were slower in finding a new word the more semantically distant it was from the previous one. And this is similar, if you think about it, to what happens in the physical domain, in the spatial environment, where it takes you longer to go to a place that is further away from you. The next evidence then is that subsequent words, participants tended to retrieve subsequent words that were semantically closer to each other. So they may say dog, cat, rather than dog, dolphin. And however, this pattern of retrieving together semantically similar items is broken down from time to time. Here is an example of a trial in which you can see the different dots being single words, and highlighted in blue are the semantically similar words, and from time to time there is a word that is semantically more different than the neighbors. And we define this word as being a switch to a new cluster of semantically related words. And this occurs around 30% of the time, and on the right you can see a visualization of what may be happening, like participants may start saying all farm animals, and then at some point there is a transition to a new cluster of semantically related animals like birds. And we further validated this definition of clusters by comparing the distances across and within clusters. And we found that indeed the semantic distance across clusters is higher than the distance within cluster. And so we can conclude that participants switched or jumped across clusters of semantically related words, balancing exploration and exploitation of their semantic memory, as it happens during spatial navigation. And the last behavioral evidence I want to show you is that within these clusters, the last word of the cluster is significantly lower in linguistic frequency as compared to the other words. So this means that you can think of this as participants running out of words to say in that cluster, and then they say the least frequent word. So to sum up these behavioral results, we have shown that similarly to how animals forage for resources in physical space, participants forage the semantic space using a clustering strategy. And these results are in line and extend previous research by Thomas Hills and Peter Todd. But for us, this was important. It was important to establish the presence of this clustering strategy to then look at the neural data. And the question now becomes, what are the hippocampal neurophysiological mechanisms that underlie the searching and finding of concepts stored in semantic memory? What I didn't say before was that our participants were epileptic patients. So we had 20 of them performing the task. And the good thing is that they had electrodes implanted directly in the hippocampus, so we can record their brain activity. And we segmented the neural activity based on word onsets, as we did before. And what happens in the hippocampus during spatial navigation is the prominence of theta oscillations. Here is an example of a rodent in its cage, and you can see that when it's still, there is very sparse and very low theta activity. But then, as soon as it starts moving, you see that there is an increase and very prominent theta activity. So this can be a correlate of spatial exploration in rodents, but also in primates and humans. And based on this evidence, we predicted an increase in hippocampal theta during mental search, drawing again on the parallelism between spatial and conceptual navigation. And to address this question, we focused on the time window before word onset, based on the reasoning that in this time window, participants are searching for and eventually finding a concept in their memory. And what we found was an increase in theta power that you can see at the bottom, indicated by the red arrow, between 3 and around 7 to 8 hertz, and between -1 second and extending up to -200 milliseconds with respect to word onset. But also, we found an increase in gamma power, highlighted there by the blue arrow, in between around 48 hertz and 145 hertz, and in the first 500 milliseconds of this time window. So this tells us that when people are searching and eventually finding a concept in their memory, their hippocampal theta and gamma oscillatory power increases. If we then average within these frequency bands and look at their time course, we can see that there is a peak in theta around 350 milliseconds. And this is very interesting because this time window was interpreted as corresponding to lexical access according to a prominent linguistic model. So this tells us that there is something about mental search and language occurring in the hippocampus. But what exactly is going on there? So going back to the behavioral data, we have shown before that we have 4 behavioral predictors of interest. So the switching behavior, the semantic distance, the temporal distance, and the linguistic frequency. And we can use these to see whether the neural activity in the hippocampus is predicted by any of these behavioral variables. And specifically, we looked directly at theta and gamma, and we used the linear mixed model to address this question. And what we observed was that theta power was significantly modulated by the semantic distance. So this means that hippocampal theta power was higher the more distant, when finding a new word, the more semantically distant this was from the previous one. And semantic distance was the only significant predictor here. Then, when we look at at gamma, we realized that semantic distance was only predicting theta because in gamma there is temporal distance as the only significant predictor. So gamma power was higher when finding a new word, the more temporally distant this was from the previous one. So we can say that different frequency bands are modulated by different aspects of mental search. And what's interesting here is that these 2 frequency bands, theta and gamma, are often reported in the spatial navigation and memory literature as being coupled through phase amplitude coupling, such that the amplitude of gamma oscillation increases at specific phases of theta. So we then asked ourselves whether there is phase amplitude coupling in theta and gamma during our mental foraging task. And indeed, this was the case. You can see there highlighted with the stars the frequencies, the couple of frequencies in which there is phase amplitude coupling, and they are for gamma between 77 and 106 Hertz, and for theta between around 5 and 8 Hertz. Then we again use mixed models to see whether any of our behavioral variables was predicting the phase amplitude coupling, and indeed, in this case, what we found was that phase amplitude coupling increases for switch transitions. So this means that the coupling between theta and gamma increases when participants change between semantically related clusters. Between clusters of semantically related words. So to conclude, we have shown several data showing that spatial and semantic foraging seem to share common mechanisms. In terms of behavior, we have demonstrated that participants use a clustering strategy in both in the conceptual space as it happens in the physical space, and this aligns with previous data by Thomas Hills and colleagues. In terms of neural data, we have reported that as it happens in spatial navigation, hippocampal theta power increases in periods of semantic foraging compared to non-foraging. And moreover, that this increase in theta power was modulated by the semantic distance between words. And this is in line with previous evidence during episodic memory task, but also during spatial navigation for spatial distance. Then, in terms of phase amplitude coupling, we have seen that there is an increase in phase amplitude coupling, similarly to what happens during spatial navigation and memory, but also that this increase in phase amplitude coupling corresponds to switches between clusters of semantically related words. And with this, I would like to conclude and thank my collaborators, and especially Simone and all the lab and you for the attention.
That was fantastic. Thank you so much. I have a question about the interpretation of the regression models you showed us trying to predict theta and gamma and so on with your various behavioral measures. Because as I understood the explanation of your behavioral analyses, all those things are correlated. So when you, for example, show a specific effect of semantic distance above and beyond the other variables in there, the way to think about that is that the semantic distance is bigger than could be explained by a switch, right? So it's like right, because it's above and beyond that switch variable that's in there, but switches are defined by big semantic distance, right? So you have a semantic distance effect that maybe is really a big jump within a cluster, right? Or similarly, the temporal distance effect is one that can't be explained by semantic distance. So there's something that's taking you longer than would be predicted by the semantic distance that I think you showed us like a 0.6 correlation, right? So I'm just suggesting that it's challenging for me, having thought about this now for 12 minutes, to think about how to interpret what looks like these very specific patterns in predicting the neural data, which are beautiful, but with highly correlated variables that are correlated in ways that's the whole premise of the behavioral analysis, right? So you've thought about this for way more than 12 minutes. Tell me how you're thinking about that.
Yeah, that's true that they are somehow all correlated. Well, first, switch, it's derived from semantic distance, but it's not just higher than the others, it's higher than the neighbors, so it's like a local measure. But yeah, the correlation thing still remains. How we address the problem of all these variables being correlated is by using several approaches, like several statistical approaches. And so the results I've showed you before were of mixed models with just one predictor at a time, plus random slopes for the predictor and random intercepts for subjects. But then we used also backward elimination, which means that you fit the full model and then remove one predictor at a time. And the best predictor and the best model remains the one that we identified with the previous approach. Then we used also the full model, which means that we fitted the model with all the predictors. So this takes into account the correlation between them. And then we used also Bayesian mixed models, which is a different way of fitting mixed models that doesn't rely on the frequentist assumption of normality of residuals and whatever. And again, we have the same results. So we have like 4 different approaches, all converging on the same evidence. And this is actually the first time I see this same results with so many different approaches. So I tend to believe that this is true.
Okay, thank you.
Sarah, and then Laurel.
Thanks for that cool talk. So I'm interested in the interpretation of this clustering effect as a strategy. So it seems like, that effect could be a function of strategy used, but it could also just be a function of the structure of the underlying semantic space, right? So in the case of spatial foraging, you can see how that would be more of an explicit strategy. You start one place, you look around, and then you decide, okay, now I'm going to go over here, and you look around, and that's like a strategy. But in the context of the semantic task, we're not seeing where people are searching necessarily, but we're seeing the output of that search process. And so if it happens to be the case that semantic information is clustered, which we have good reason to think that it is, then why would this clustering effect be interpreted as a strategy and not just a function of the underlying structure?
I see. Yeah, that's true. We didn't control the structure of the semantic space because this is a naturalistic space like animals, cities, and professions. Maybe the way to address this question would be to design a new experiment with a controlled environment and then check whether it's indeed a strategy or a reflection of the structure of the space. Yeah, that's something we can do next. Thanks for the question.
Thank you. It was really interesting. I have another interpretation question. So I like your account in terms of semantic foraging, but I think the story for me would even be stronger if you were to show that when you looked at other regions that we know, and this is related maybe to Sarah's question, other regions that we know also represent semantics that, unlike the hippocampus, you would not predict to show this effect of clustering and switching, so that, in other words, it's really this sort of something special about the way the hippocampus organizes search behavior that is mechanistically in common to both these spatial and these semantic tasks. And the other thing has to do, sort of echoing the comments about everything being correlated, is that, with increased cognitive effort, as things get harder and you get stuck, you would expect that everything's going to activate and start firing together. So I would also love to see that this is not merely an effect of something getting hard and then everything starts to ramp up.
I see. Well, thanks for the questions. Regarding your first point, these are patients, so we cannot record from the whole brain, and the implantation is patient-specific. What we have and we can look at is the lateral temporal lobe, because the electrode goes from, well, yeah, I'll just show you the data. So the electrode goes from the lateral to the medial part. So we have the lateral temporal lobe, and when we look at that area, we can see that the time frequency is quite different in terms of both the frequencies that are activated and also how they are activated, and also the temporal dynamics. And so this also, yeah, the lateral temporal lobe is somehow related to semantics, and something different is going on there. And your second point was about correlation and effort. Yeah, how would you control for that, though? I mean, it's probably a completely different experiment, I guess, like control for effort. I see. I see. Yeah, I mean, if we can find a measure, maybe we can use that as a covariate in our models and see whether it explains some variance. Thanks.
All right. Well, thank you so much.
Thank you.